My name is Matt Lawn. I'm an associate professor at the University of North Carolina at Chapel Hill, and I am a member of the steering committee of the Pediatric Trials Network. So we recently published an article on innovative trial design in pediatric therapeutics, and children really are a special population, and they're very challenging to do trials in children, particularly infants, particularly preterm infants. And this is because there are challenges with consent, because families and parents don't always want to consent. There are challenges in the amount of blood that we can take from babies and from premature infants. For example, a baby who weighs 500 grams or about a loaf of bread has a circulating blood volume of about 3 to 4 tablespoons. And so we can't take much out. And so it really requires a lot of expertise in order to perform these studies. So we've done a variety of things in order to improve recruitment into trials, into pediatric therapeutic trials in the Pediatric Trials Network. We've worked with the lab to try to develop assays for the molecules that are very sensitive, so it requires a very small amount of blood, usually in the range of 200 microliters. So that's 10-fold less, maybe 15-fold less than what we would do in an adult. And we are also working to sort of move the field forward by using dried blood spots, which is a technology whereby we use even less blood, 10 to 20 microliters, on a dried blood spot. And it has lots of advantages because it doesn't require any special processing, it's very low blood volumes, and it can be left out at room air. There doesn't need to be a freezer, for example. The other things we've done is to take a few samples from more babies or from more children, and we call that sparse sampling, so that we don't take multiple samples from the same person or from the same child. Along with that, in order to analyze that, we use special advanced pharmacokinetic techniques, advanced modeling called population pharmacokinetic modeling, where we can take all of the samples from the babies at different times and put them together, and the infants act as if they're a larger population than they actually are. Using those techniques in the Pediatric Trials Network really has been beneficial to the infants and to the children, and has made it for a more efficient trial design. So we're using these methods successfully in almost all of the studies that we do. Use one or more of the techniques that I just mentioned. For example, metronidazole recently finished in under a year using sparse sampling and dried blood spots, as well as something called scavenge sampling, which is taking blood from the lab, the clinical lab. Lots of these babies have blood draws and chemistries that are done, and there's always blood left over. And so instead of discarding that blood, we have our coordinators at the different sites go down and collect that blood. And then in almost all of them, we're using population pharmacokinetic modeling.